Hello, welcome to the RNA-seq data analysis platform powered by Genialis. When you have obtained your raw RNA-seq data, you want to dive right into analyzing your data. We have developed a web app to make this process easier than ever. Here you can run validated bioinformatics pipelines to process and explore your data. So let us go ahead and get started. If you already have an account, you can just sign in. If you do not, you can easily register for one here. Alternatively, if you are waiting for your data to arrive from the sequencing core, you can enter as a guest and explore some of our sample data. So let's sign in. After logging in, you arrive at your personal homepage where you have some of your latest data and data highlights. We have prepared a self-guided tour for you. For the purpose of the demo, we will be using published data on the effects of the drug JQ1 on triple-negative breast cancer cell lines. The reference data is linked here. So let us see how you can upload your data and run analysis. This can be done via the recipes page found here in the navigation bar. The first step is to define the experiment. Here you can specify your organism, if you choose to remove the adapters, the assay types, etc. Then you can upload your raw or compressed FASTQ files via drag drop or the easy clickable interface. While your sample is getting uploaded, you can check it here. You can even change the sample name to something more user-friendly. Once your samples are uploaded, you can start your analysis by clicking here. With a single click, you can run the analysis from trimming to alignment to quantification. The status icon tells us that the sample is running. You can hover over or click on the icon to see the current status. While the analysis is running, we can see that the results table is getting populated. Everything on this table is clickable. For example, you can check the QC report of your sample. If you click on the sample name, you come to the sample details page. Here on the left, we give you the analysis history. We do not want you to worry about the bioinformatics, but you should always know what is going on. So here we list each tool that was used, all the inputs and outputs. Also in this page, you can annotate your sample. Now let us examine some data. Let me search some sample of interest. I can select all the samples and using this icon, add them to the sample basket. Let us now navigate to the interactive visual analysis page. Here you have several tabs to perform various exploratory analysis. The first question one might have is, are my replicates consistent? Are my samples and controls distinct from one another? We can check that in the sample comparison page, where we can see if my samples are clustered as expected. Let us now check the expressions of some genes of interest. You can select your genes here and the expressions will be loaded. We can see that the common cancer markers do not vary between treatment and control. So next, we would like to find out which genes are differentially expressed. You can calculate a differential expression analysis by clicking here, where you can specify your case and control samples. I am selecting a pre-calculated analysis. In the volcano plot here, you can see the upregulated and downregulated genes in response to the drug. So let us select the genes that are downregulated and then add them to the gene card. Here. You can also save these genes as a new gene set. In the When Viewed tab, we can identify the genes specific to a certain group. For example, let us create a Venn diagram and see which genes are specific to the susceptible cells. 42 genes are specific to the susceptible group. The gene ontology enrichment analysis is also performed. Now, let us find out if any of these genes show drug sensitivity. For that, we will explore the heat map to identify any gene that shows striking visual pattern. The logarithmic scale will provide easy visualization and we will display the gene names. Two genes stand out, CXCL1 and NNMT. Both are known to be associated with cancer and can be promising target for the drug JQ1. At each stage of your analysis, you can export the data and the figures using this icon here. If you want to analyze the data in your local machine, you can easily do so. First, you need to select the samples that you want to analyze and then click on this download icon. Then you can select the files that you want to download. You might also want to share the data with your collaborator. You can easily do so using this icon. We support granular permission on all data. As long as your collaborator has an account, you can share the data with them. This is Genialis platform powering RNA-seq data analysis.